Why are scientists so interested in changing a cell's identity in the first place?
Because a cell's identity controls what it can do, and changing it could help us study disease, repair tissues, and understand aging. Most cells are locked into a role, like skin or muscle. The Yamanaka factors became famous because they showed that, under the right signals, mature cells can be pushed back toward a more flexible state in the lab.
So what exactly are the Yamanaka factors?
They're 4 proteins, OCT4, SOX2, KLF4, and c-Myc, that act like master switches for gene activity. When introduced into certain adult cells, they can rewind the cell's program toward a stem cell-like state called an induced pluripotent stem cell, or iPSC. Pluripotent means it can potentially become many cell types, though the process and outcomes depend on conditions.
How do 4 proteins actually persuade a mature cell to reset its identity?
They bind DNA and reshape the cell's gene network, turning on stemness-related genes and quieting specialized ones. Over time, they also influence epigenetic marks, like chemical tags on DNA and proteins that affect how tightly genes are packaged. Think of it as loosening an old instruction manual and activating a more flexible one. It's gradual, imperfect, and many cells don't fully reprogram.
What has this discovery actually enabled scientists to do?
It let researchers make patient-specific iPSCs, which can be turned into neurons, heart cells, and other types for studying disease and testing drugs in a dish. It also supports regenerative medicine research, because you can explore how to replace damaged cells. Separately, partial reprogramming is being studied as a way cells might regain youthful features, but that's experimental and not a proven therapy.
What are the biggest risks or downsides when you try to reprogram cells?
Control is the big issue. If reprogramming is incomplete, you can get abnormal cells, and some factors, especially c-Myc, are linked to cancer when misregulated. Delivery methods can also disturb the genome. Even with safer techniques, you must verify cell identity, purity, and stability. Ethically, there are concerns about misuse, unproven anti-aging claims, and equitable access if therapies ever mature.
If someone remembers only 3 things about Yamanaka factors, what should they be, and where is this heading?
First, they're 4 gene control proteins that can reset adult cells toward iPSCs. Second, they transformed research by enabling patient-specific cell models and new drug testing approaches. Third, it's powerful but risky, so control and safety testing are central. Looking ahead, the vision is more precise reprogramming, maybe partial, timed, or tissue-specific, plus better delivery and monitoring, before broader clinical use.